Welcome to Biology My Passion. I am Soumya Harikrishna. We are learning the chapter Biomolecules. We dealt with the analysis of molecules. We also learned biomicromolecules and biomacromolecules. Now we are coming to the last part of this lesson. It is about enzymes. We have heard of enzymes. In class 10, you learned about 4 enzymes which are involved in digestion. And also in respiration chapter this year, we are learning about carbonic anhydrase enzyme. What are enzymes? Enzymes are basically biological catalysts or they are organic catalysts. What is meant by that? Catalyst is a substance which speeds up the rate of a reaction without involving in the process. If you remember, in class 10 you learned a lot of equations, but only in one equation, that is hydrogenation of a vanaspati, you learned nickel or palladium as a catalyst. Rest of the reactions we did not mention catalysts. That means all reactions do not require catalysts for their function, but when the same reactions are happening within our body, enzyme is a must or organic catalyst is a must. So every biochemical reaction in our body is happening with the help of certain catalysts, and these catalysts in our body are called enzymes. I will give you an analogy to understand this. For example, I am a teacher. I am teaching class 11 and 12. Okay. So when I am taking a batch in 11, in our school it is usually 11th and 12th, a continuous class. The same teachers handle the batch till they pass out. Like 9 and 10 same teacher, 10 and 12, 11 and 12 same teacher. So I get a new batch in 11th. I will be teaching them and preparing them to get ready for the 12th board exam. So when I get them in my hand, they are like a 10th standard knowledge small babies. So I am preparing them, not only me, the teachers, we prepare them to reach the level of facing the board examination, and they pass successfully and they go for professional courses. Mostly they go for medicines or engineering or abroad some courses and all. So am I going with them? No. I joined them in 11th, till 12th I will be with them or we will be with them, and after that we send them out. Okay, they are ready to go to the world of life. We come back to another new batch, again we are continuing the reaction. Enzymes are like that. They convert a substrate or reactants into product, but they don't get involved in that reaction. At the end, products are formed. The enzymes will be ready or available for another set of substrate to catalyze the reaction. Understood? Now, what are these enzymes? Are they similar to the catalyst outside? No. Here these enzymes are biological or organic in the sense that they are proteins. We just learned about proteins in the previous video. The proteins act as enzymes except for 1 or 2 exceptions, because RNA, RNA is a nucleic acid that can also act as enzymes in certain reactions. Rest all enzymes are proteins. That means mostly enzymes are proteins. So we make a statement, all proteins are not enzymes, but all enzymes are proteins. But as I told, there is an exception. The ribosome is one exception you can remember, which is actually a nucleic acid or RNA. Then another thing, when the enzymes are produced, if you remember last year when you learned digestion, we learned that there is an enzyme in the stomach, pepsin, for digesting protein. But we learned hydrochloric acid function as activating this pepsin. That means pepsin is present in an inactive form called pepsinogen, and then it will be converted into the active form called a pepsin. So most of the enzymes are secreted as a proenzyme, means inactive enzymes. They are also called the zymogens. Then proteins are the basic nature of enzymes that we mentioned, and also enzymes are very specific in nature. So if there is an enzyme for catalyzing a particular reaction, that enzyme cannot catalyze another reaction. So for every substrate, substrate means the substance in which a reaction starts or reaction happens. So for every substrate, there is a specific enzyme to catalyze that. No other enzyme can take up the role and continue the reaction. Okay. Another example, when we talk about specificity of the enzyme, is the teachers. Again, I told about teachers in the previously. The same way, I am a teacher who is teaching biology. Okay, up to 10th standard we can handle almost all subjects. But when it comes to 11 and 12, we are very comfortable with the specialized subject. So I am teaching biology. I am very specific in my subject. For physics, another teacher is there. For chemistry, another teacher is there. For math, another teacher is there. For psychology, physical education, all subjects have got separate teachers, or computer science, their subjects are different. So we are specific in our nature. So my class, I can teach only biology. I cannot go and teach physics or chemistry or anything like that. So we are very specific in nature. And also, if I am absent, maybe a physics teacher will go to class, but she is not going to go and teach biology for them. She will teach her subject only. That's what specificity. Or I cannot go and teach in commerce stream or humanity stream. There the expert teachers are there to teach them, right? So that is the condition. So enzymes are very specific. I am very specific to my science students who have taken biology as their optional subjects. So that's called a specificity of enzymes. They can catalyze only a particular substrate. And they are thermolabile. That means enzymes are mostly very sensitive to the temperature. Means at high temperature and low temperature, their efficiency is affected. They all work in a very optimum temperature suited for that particular enzyme. That means mostly in our body, we know our body temperature is 36 to 37, average we say 37. So our enzymes are in that range. So most of the land organisms have the range of 25 to 35 degree, but there are bacteria living in hot thermal vents. Their average temperature is 100 degree. So they are most thermostable enzymes, but other enzymes are mostly thermolabile. We will learn about the effect of temperature towards the end of this session. Usually enzymes, last class when I taught you about the tertiary structure, I told there are certain crevices or pockets to which the substrate can bind. So here also the enzymes have the structure so that the substrate can bind to it. So it forms enzyme substrate complex and then it starts the catalytic reaction. Now let's see how can we talk about the rate of reaction. You know that chemical reactions happen, but how can we decide the rate of that particular reaction? By taking into consideration the time also as a factor. So we can say that rate of a reaction is delta P by delta T. Delta P means the difference in the product formation. That means the amount of product formed. For example, suppose 100 molecules were there in the beginning. Now it has become 1000 molecules. So 900 molecules is the delta, means difference, right? At the rate, time also we are telling, only then we call it as rate of a reaction. So at a particular time, maybe 2 minutes or 3 minutes or 4 minutes or 1 hour or 2 hours or 3 days, like that. So this gives us the rate of a reaction. But generally it is observed that whenever an enzyme or a catalyst speed up a reaction or involved in a reaction, the rate of the reaction is extremely high. That is, it becomes many-fold. I will give you one example to understand that. There is a very simple example we study in respiration, that is carbon dioxide combines with the water to form carbonic acid. This usually happens in nature also. Do you know when is this reaction happening in nature? During rain. So when water is coming down, it mixes with some carbon dioxide in the atmosphere to make carbonic acid. So rainwater is slightly acidic, we can say. But for that reaction, there is no enzyme or enzyme we cannot talk about. For that, no catalyst is required, right? So the rate of the reaction is 200 molecules forming per hour. So it's a slow reaction. Only 200 molecules of carbonic acid forming in 1 hour. But the same reaction happens in our body during carbon dioxide transport in our blood. So during that time, we have uploaded the breathing chapter video, so you can refer to that if you want to know. So there, in that case, H2CO3 is forming because carbon dioxide when transported in the bicarbonate form, we are using this equation. But in that case, we are adding one enzyme to this scenario, that is carbonic anhydrase. So this enzyme when it is coming into this picture, the rate of the reaction becomes 600,000 molecules per second, not hour, second. Every second 600,000 molecules are being formed. So here the rate is almost increasing 10 million times when we are adding enzymes to the reaction. Now, the rate of the reaction can also be called as velocity of the reaction if we mention the direction also. When we talk about temperature, every 10 degree rise or decrease in the temperature can affect the rate of the reaction. For example, for every 10 degrees Celsius increase, the rate of reaction can become double. The same way, every 10 degrees Celsius less in temperature or decrease in temperature, the rate of chemical reaction becomes half, but within that specific time temperature limit. Okay, whenever you are watching my classes, instead of simply watching like a video, you take your textbook with you and also a pen or a pencil or some notepad or notes where you always refer to, or in sticky notes you can write the points and then keep it there corresponding to the textbook, because textbook has lot of space to the side, or you underline the points in the textbook. Any extra point is there, you can take it there itself. So simply if you watch, you will forget. None of us have super brains, so we tend to forget. So if you want to learn, means you make notes out of it, because I usually give all the points on the boards. At least these points if you can take down, then that will be helpful for you to remember the points one after the other. Clear? So please keep a note always whenever you are watching my videos. Okay, we learned that enzymes can bring about a very drastic change in the rate of reaction. How can enzymes do this? We are now going to discuss that. So whenever we talk about enzymes, I told about they are acting on certain chemicals. So the chemicals in which a reaction happens is called a product. That means the chemical which gets converted into product. In chemistry we call it as reactants. Okay, so that's called a substrate. Now, how many molecules of substrates are converted into products per minute? That is called the turnover number of an enzyme. So that shows the efficiency of the enzyme. If turnover number is high, enzyme is very efficient. Then active site. Every enzyme, I told the three-dimensional structure I showed you in the previous video. In that some crevices are there. To this only the substrate can bind. So that is called the active site of the enzyme. For example, if you draw an enzyme like this, okay, consider these two are the active sites of the enzyme. To that, a substrate will come and bind. Suppose the substrate shape is like this. Now what happens? It can come and bind here. Right? So this is usually called a lock and key model. Why? Because we already told enzymes are very specific in their nature. So when they are very specific, how a lock can be operated by a particular key, the same way one enzyme can bind to a specific substrate only. Right? So that's called a lock and key model. So why? Because this active site has that configuration to which only a particular substrate can fit. The active site of the enzyme is otherwise called the substrate binding site. So whenever there is an enzyme binding with the substrate, so this is enzyme, it is binding with the substrate, an enzyme substrate complex is forming, and there happens a transition state. Transition means what? The substrate is getting converted into product. So enzyme will be holding the substrate. Suppose this is the substrate, my hand is the enzyme, it is holding the substrate. While holding it, it will make necessary changes in the bonds of this substrate structure so that it will be converted into product. So this change happens during a time that is called a transition state. Enzyme and a substrate, they work together for this transition state. But for this substrate to reach the or become the product, it needs to attain a particular energy level. Suppose if you plot it as a graph, you know this is the, suppose the potential energy of the substrate is at this level. This is the energy of the substrate at the current time, and we are making the enzyme bind to that substrate. So the energy level is going up when now I am showing without enzyme first. So the substrate, any reaction to happen, the substrate or the reactant should reach a transition state. Okay, so that is called the activation energy. Now at this energy, if it reaches, it will break into the product or form the product and come back as product. Okay, so every substrate has to reach this activation energy during the transition state to get converted into the product. So that makes the lag in the time, because the time taken for reaching that energy level from this level to almost this level is very high. But what is an enzyme doing? enzyme will bring this activation energy to a lower level. Suppose you are doing high jump, okay, and you are very comfortable jumping up to 1.2 meters. The crossbar is kept at 1.5. So when you are jumping, you are not reaching that height. Okay, so what is your coach doing? Your coach will bring down the crossbar to 1.2 so that you can easily cross that. So after practice, he will gradually increase later. So when you are not able to jump to that height, he decreased it so that you could climb over or you could jump over that, right? So the same way, enzymes are like your coach. It is bringing down the actual rate below. That means instead of reaching this level, it is keeping the activation energy to a lower level so that the product can form easily. No need to reach this gap. Okay, so enzyme is decreasing the amount of activation energy. So within that time, by reaching, it can make lot of products. So this is how enzymes are making the products.